Okay, so let's just continue with our discussion, and we will discuss two topics. One is antigen antibody interaction, and the second topic that we'll discuss now is antigen processing and presentation. Okay, how is antigen processed and presented? We already briefly discussed this. Now we are looking at the intricate details. Okay, what are the molecular mechanisms involved here? Okay, first let's start our discussion with antigen antibody interaction. Okay, so if you look at the antigen antibody interaction, the antigen and antibody, when it forms complex, so basically the antigen and antibody, we know the antigen has epitopes on its surface and the antibodies have paratopes on their surface, and you see the interaction happening between the epitopes. So this is what we saw, right? So this is what we saw. So you see the epitopes and the paratopes actually interacting. So the epitopes on the antigen and the paratopes of the antibody, those are the regions which actually interact. So if you look at this portion, just like how you saw the active site of an enzyme. If you remember the active site of an enzyme will form a pocket, okay? And into this pocket, the substrate will come in. So there's a lock and key model, okay? Just like, you know, how for every specific lock, there is a specific key with which it opens. In the same way, for every specific paratope, okay, which basically forms a pocket, okay, you have a specific antigen which will actually fit into it, or specific epitope which will actually fit into it. Okay, now if you look at the interactions happening here between the paratope and the epitope, you will see that none of the interactions are actually covalent. So if you look at the interactions between the paratope and the epitope, so the pink one is your antigen. The pink one is your antigen, and this is your antibody. Okay, so you see the interactions are, all the interactions that occur between the antigen and the antibody are non-covalent. You see certain hydrogen bonds forming, you see certain ionic bonds forming. Okay, ionic bonds are electrostatic bonds between two different charges. Then you see hydrophobic interactions between hydrophobic groups. Okay, you see van der Waals interactions because they are very close to each other. Okay, non-specific weak electrostatic forces that occur between neutral molecules when they are closer together. Then there are certain weak electric forces, okay, between neutral molecules also, and of course the same ionic bond shown here again. Okay, so you see all these are non-covalent, so they can be easily broken, and the antigen and antibody can be easily separated. In other words, the antigen antibody interaction is reversible. Okay, just like how the antigen and antibody are coming together, they can be easily separated into antigen and antibody again. Okay, so if you want to separate that, so let's say, let's take this, let's say this is the only bond that is holding them together. I'm just giving you an example. Let's say this is the only bond holding these two together. Just ignore the everything else. Let's say these are the two bonds which are actually involved in this interaction. So this amino group has a positive charge, and this amino group will also have a positive charge. Okay, so what will happen if I increase the pH of, let's say this antigen antibody is in a tube, and let's say the pH now is 4. The pH now is 4. I'm giving you an example. Let's say the pH is now 4, And at 4 pH, this amino group has a positive charge. This amino group also has a positive charge. Just ignore all the other bonds. Only these are the two bonds that are existing between the antigen and antibody. If I change the pH, then immediately the charge on this, if I increase the pH, at some point, the amino group will lose the proton. This amino group will also lose the proton, right? So the attractions will no longer be there, and the antigen will separate from the antibody. Okay, so by changing the conditions, okay, we can actually separate the antigen and antibody complex. Okay, in the same way, just instead of ionic bonds, just imagine they are bound only by hydrogen bonds. Okay, none of the other things are there. Okay, none of the other things are there. Then these hydrogen bonds, can be easily broken if we increase the temperature. So if you take this antigen antibody mixture and heat it up, then immediately these hydrogen bonds are broken. You see how we discussed in the DNA, right? In the DNA, the two strands of the DNA are held together by hydrogen bond, two hydrogen bonds forming between A and T, and three hydrogen bonds forming between G and C. Okay, those can be easily broken, and the two strands of the DNA can be separated if you start heating the DNA sample. In the same way, the antigen and antibody can be separated because they are held together only by hydrogen bonds. Okay, so changing the conditions of the antibody and antigen complex, okay, changing the conditions of the antigen and antibody complex, you can basically separate the antigen and the antibody. So antigen antibody reactions are reversible. Just like how they're coming together, they can be easily separated from one another because they are interacting only with non-covalent interactions. Okay, there are no covalent bonds involved here. Okay. Now, they are reversible, and you have this equation. Okay, this is antibody antigen, the concentration of antigen antibody complex by individual. Okay, so here this is what we call the affinity constant. Affinity constant. Okay, this we call the affinity constant, and okay, these are molar concentrations of free antigens and antibodies. Molar concentrations of free antigen and antibody. This is for antibody, this is for antigen, and this is for complex. Okay, for the antigen antibody complex. Okay. Now, the values of Ka or the affinity constant are usually in the range of less than 10 to the power of 5 mole inverse, okay, mole inverse to 10 to the power of 12 moles. Okay, so usually in this mole inverse. So usually in this range, the affinity constant is 10 to the power of less than 10 to the power of, 10 to the power of 5 mole inverse to 10 to the power of 10 to greater than, to greater than 10 to the power of. So usually in that range is where you find the affinity constant. And this affinity constant is affected by, as we have seen just now, by temperature, by pH, and also by solvent. Okay, so if you put this in a highly hydrophilic solvent, then the hydrophobic interactions will be much higher. In a hydrophilic environment, the hydrophobic groups tend to bury themselves inside, so the interactions can be stronger. Okay, so those factors actually determine the affinity constant, the temperature, pH, and the solvent, nature of the solvent. Okay. Now, there are two terms as far as antibody and antigen interactions are concerned. One is called affinity and the other one is called avidity, AVI. Okay, now affinity is the strength of interaction between, okay, this is the definition, strength of interaction between, okay, a single site on an antigen and antibody. That means if I go back to this example, okay, if you go back to this example, okay, what is the strength of, okay, what is the strength of interaction between this epitope and this paratope? This epitope and this paratope. If I take this single interaction, okay, strength of interaction between single site on an antigen and an antibody. If I take this single one, what is the affinity here? That is my, what is the attraction here? What is the strength of attraction here? What is the strength of interaction here? That is the affinity. Okay, that is the affinity. Now, there's the other thing which is avidity. Okay, so avidity basically by definition is overall strength of antigen antibody complex. Okay, overall strength of antigen antibody complex is your avidity, overall strength. Now, what do I mean by that? So, let's say this is your antigen and this is the antigenic determinants or epitopes on the surface of the antigen. Okay, now this antibody, each antibody we know have, okay, we know have a valency of 2, at least a valency of 2. Okay, so when it starts to cross-link, you see it's actually binding two different, with one valency it is binding this, and with one valency it is binding this, and this antibody is binding with one valency with this, one valency with this. So the entire thing is actually forming a complex. So when it is actually forming a complex, the overall strength of all these interactions, is avidity. Okay, it's like this. If you take a single stick, okay, it's easier to break. Okay, but if you take multiple sticks, okay, and form a stack, it becomes very difficult to break. Okay, so avidity is always higher because there are multiple interactions involved here, just like how we saw here. If you take just one interaction, that's your affinity. Okay, the strength of interaction between this paratope and this epitope is affinity. But the overall strength of this complex, is avidity. Avidity is always higher than affinity. Okay. Now, the avidity will depend on 3 factors. Avidity will depend on 3 factors. The first one is antibody epitope affinity. Okay, antibody epitope or paratope epitope affinity. Okay, how it is the individual strength of attraction. Okay, how good is the individual strength of attraction? Then the valence of antigen and antibody. Okay, we know that antigens are multivalent, even the antibodies can be multivalent. Okay, in the case of IgG, they're just divalent, but if you take IgM, they have a valency of 10, multivalent, and if you take IgA, they have a valency of 4. Okay, so avidity will depend on the valency of the antigen and the antibody, valency of both antigen and antibody. Then the third thing is structural rearrangement of the interacting parts. structural rearrangement of the interacting parts. Okay, whatever parts are actually involved in those interactions, whether they can change their structure or not. Okay, if they are very rigid, okay, if they are not flexible enough, then you don't see a lot of interactions actually happening. Okay, but if they are more flexible, then they can twist themselves and turn themselves and basically cause or end up in several different kinds of interactions. So these are the three factors which actually influence the avidity of an antigen antibody interaction. Okay, now an antigen antibody reaction is usually specific. There is specificity in antigen antibody reaction. That is, a paratope will actually, a single paratope will interact with only one kind of an epitope. Why? Because there are certain groups that have to form non-covalent bonds, and only when you have this particular group, I'm just giving you an example, okay, opposite to this, and only when you have this particular opposite, so these can come together only in one particular combination. So when you have this antigen and this antibody, only when the epitope of this antigen, the epitope paratope of this antibody, when they come together, these are the interactions formed, and that is how the antigen and antibody complex are being stabilized. Okay, so there will be a specific epitope and a paratope which can come together. So the antigen, in other words, the antibodies are highly specific. They bind only to the specific epitope of a antigen. Okay. However, we already discussed cross reactivity. There are a few instances. There are a few instances where antibodies generated against a particular antigen can interact with a different antigen. Why? Because they might share the same kind of epitopes. They might share certain epitopes which are common. We discussed about the example of smallpox and cowpox. Okay, so that is about the antigen antibody interactions. So next we will look at the antigen processing and